So I got started talking last time mostly about DNA structure, and today I'm going to talk about some DNA replication. And I've got some videos and so forth I'm going to show you. So not all the talking will be done by me today. Hey. All right. So I want to think about DNA structure and as an introduction into talking about DNA replication. Everybody hears and knows the term that DNA strands are anti-parallel. And that has a meaning. And that meaning is, well, this one goes this way, this one goes this way. And that's anti-parallel. What you need to know, of course, as I'm sure you all probably do know, is that DNA strands have a polarity associated with them. They have, and all that means is that they have different ends. Okay? So we can tell the ends apart. The molecule is not symmetrical. And if we think about an individual nucleotide, we can think about that structure there of, let's say, AMP. Okay? If we think of AMP, we think of the fact we've got a sugar down at the bottom, we've got a base up on one side, and we've got a phosphate up on the other side. The phosphate is linked to carbon number 5. Okay? So the phosphate's on carbon number 5. All the action that happens in nucleic acids, with one very minor exception, or with one exception that I'll talk about later in the term, occurs between carbons 5 and 3. So if I think about carbon 5, it's got a phosphate on it. I think about carbon number 3, it's got a hydroxyl on it. When I grow a nucleic acid, the nucleic acid grows by the next nucleotide coming in and a phosphate linking to that hydroxyl on number 3. So now I can picture in my head I've got two nucleotides. I've got a top one, I've got a bottom one. The top one is where that phosphate was. Notice the phosphate on position 5 has not changed. But the carbon number 3 that was previously a free hydroxyl is no longer a free hydroxyl. It's joined to a phosphate, which is joined to the next nucleotide. So what I've seen is that the hydroxyl on the original carbon number 3 is gone, and now there's a new carbon number 3 on a new nucleotide that has a free hydroxyl. So what we're seeing in that sort of mental image is that the strand is growing in what we call the 5' to 3' direction. All new nucleotides will join a previous 3' hydroxyl. Okay? So if we extend this out a long ways, now we think, okay, well, here's a 5' end. Here's where that original phosphate was. Here's where the last hydroxyl is. So I've got 5' end, I've got 3' end. And when I look at nucleic acids, they arrange themselves anti-parallel, meaning that this other strand that it pairs with has a 5' end and a 3' end, and they're arranged like this. So that's what the anti-parallel means. Another thing about the structure of nucleic acid, double-stranded nucleic acids that we have to think about is the orientation of everything. I didn't point that out to you last time. Okay? The orientation of everything. We think, okay, well, here's a strand on top, here's a strand on the bottom. But of course, we know it's a coil, right? And that coil is arranged in not a random way. It's organized in a regular way. But what we learn in thinking about the structure of DNA is very similar to what we learn in thinking about the structure of a protein. When we thought about the structure of a protein, we thought about the fact that the hydrophobic amino acids arrange themselves on the inside of the protein. The hydrophilic amino acids were on the outside of the protein. Hopefully everybody remembers that from last term. Okay? When we look at a DNA double helix on the outside, okay, so we've got an arrangement that I'll show you here. Yeah, it's not a very good arrangement. Not a very good arrangement. Well, this will work. All right. We've got an arrangement of, on the outside, we have linkages between sugar, phosphate, sugar, phosphate. That's those linkages I was describing to you earlier. The phosphate coming in and joining to the next sugar. The sugars and the phosphates are on the outside of the helix. They are hydrophilic. The base pairs that we think about with DNA, A pairing with G and C pairing with A pairing with T and C pairing with G, are arranged on the inside, and they are relatively hydrophobic. Okay? So DNA has to exist in an aqueous environment, and it arranges itself so as to do so. Hydrophilics on the outside, hydrophobics on the inside. Now, it's not quite as pronounced as it is in proteins. In proteins, you can have those amino acids literally buried in the structure. And in DNA, you don't have that. But the general arrangement of the chemical groups in DNA is not that different from the general arrangement of the chemical groups in a protein. Okay. Well, now we've oriented ourselves with the structure of DNA. Let us start considering the replication of the DNA. So replication of DNA is a pretty phenomenal thing, and we are still learning a lot about that. The pioneering work done in determining the structure of DNA was done by a man named Arthur Kornberg, who was a researcher at Stanford University, who began looking for enzymes that helped to duplicate DNA. He started doing this in the 1950s, not too long after the structure of DNA itself was determined, because he knew that everything else in the cell was essentially catalyzed by an enzyme. It made sense that DNA replication would also be catalyzed by an enzyme. And so he began searching for enzymes that would replicate strands of DNA, and he found some. The first one he found, he named Dna Polymerase I. And the structure of Dna Polymerase I, we now know today, looks like this. And Dna Polymerase I is probably the most studied DNA replication enzyme on the face of the earth. Okay? It is an E. coli enzyme. In fact, all the enzymes I'll be talking about in the first part of the talk are all E. coli enzymes. E. coli being the bacterium in our guts, very easy to grow up and make a lot of it. Now, if we look at the structure of this guy, we can see it, and we can think about the structure as something like my hand. We have a thumb. Okay, well, actually, they've got to orient this way, but you can look at it either way. A thumb, and we've got fingers, and the palm is where the action is. The palm is where the action is. Meaning, this is where the reaction is, where the DNA is held, and where the catalysis occurs to elongate the DNA strand. Okay? Now, Dna Polymerase I has some interesting properties. And when they were first discovered, it was a little unusual, it was a little surprising. But these properties that Dna Polymerase I has are found in many other DNA replication enzymes. Not all, but many. Okay? So I'm going to describe them to you. It turned out that Dna Polymerase I had 3 different catalytic activities. The first catalytic activity was they found that it had was the one they were looking for, and that was the one that DNA, it polymerized nucleotides according to the base pairing rules. A went next to T, C went next to G, etc. So DNA polymerase I, like all DNA polymerases, joins an incoming nucleotide to a previous nucleotide. It makes DNA in the 5' to 3' direction. And in doing so, it creates what we call phosphodiester bonds. Phosphodiester bonds. That simply means that when we're done with the thing, we have a phosphate, and on either side of it, we have a sugar. A phosphodiester bond. All DNA polymerases make phosphodiester bonds because that's how you put together the strands. If it couldn't do that, we wouldn't have a polymerase. All RNA polymerases make phosphodiester bonds. This wasn't surprising. One surprising thing that he found was that DNA polymerase could not start a DNA strand on its own. If you gave that DNA polymerase a single strand of DNA with a 5' end here and a 3' end here, you would think, well, it would start at the 3' end and it would replicate in an anti-parallel fashion, 5' to 3' back here. But when you put simply a single strand and DNA polymerase I and 4 DNTPs, that is DGTP, DCTP, DATP, and DTTP, nothing happened. Okay? Nothing happened. Now, something was missing. And the something that was missing turns out that DNA polymerase, like all DNA polymerases, can only extend if there's a previously base paired little piece on there called a primer. So now we have to think about it looking like this. Okay? Here is my 5' to 3'. Replication's got to come back this way, and I'm going to use my fingers here to indicate a primer, which is a short stretch going 5' to 3', pointing back this direction. All DNA polymerases require a primer to start DNA replication. So if I just give it a single strand, nothing's going to work. I have to have something else on there. This is a problem. Because if a DNA polymerase can't start synthesis without an existing piece there, how does the existing piece get there? Okay? I heard somebody say the magic word. And the magic word is that cells use RNA. RNA polymerases are capable of starting with that single strand and starting and making a short segment called that primer. And so that when I'm done with an RNA polymerase doing that, and by the way, there's an enzyme that does that. It's called primase, P-R-I-M-A-S-E. Primase will come along, and it will put that primer on the end so that now I've got something that DNA polymerase can extend off of. A DNA polymerase can use an RNA as a primer. In fact, in most cells, that's exactly what happens. In fact, in all cells, that's exactly what happens. Some viruses have some unusual schemes, but all cells use RNA primers for their DNA polymerase. Okay, everybody with me so far? Okay, so that was a surprising activity. I'm going to say more about that later. But I want you to remember that we have to have a primer on there. When we do DNA replication in a laboratory, we can cheat. We can make a primer. We can actually make a primer chemically. We can go to a machine and say, give me a primer that has this sequence such that it'll form base pairs out here. And that primer doesn't have to be RNA, it can be DNA. Cells don't have that luxury. Cells have to go through RNA in order to start the replication. Okay, now, I said DNA polymerase 1 had 3 activities. Okay? The second activity that they found in DNA polymerase 1 was one that after they discovered it made a lot of sense. Okay? At first it seemed a little odd. It's an activity that destroys a strand of DNA. So you've got one activity that's making a strand, and you've got another activity that's working in the opposite direction, destroying that same strand. Seems a little odd. It's kind of like creating work for yourself, right? If your job is to make something at the factory, okay, and then when you're done making it, you destroy it, you've always got a job, assuming the company doesn't run out of money with your destroying the thing. We have to make 50 cars today. Okay, we make 50 cars, we destroy them. Tomorrow we have to make 50 cars again. Well, I'm being silly, and that's not what happens with this activity. What this activity does is it provides a way of checking to see if replication has proceeded properly. Okay? This activity has a name. It's called a proofreading activity. And what the proofreading activity does is the same thing that a proofreader of a magazine or a newspaper would do. They look for typographical errors. A proofreading activity looks for typographical errors. Okay? Now in DNA, we've got a 4-letter alphabet. The typographical errors are, if I go along and the DNA polymerase is doing its thing and it goes A next to T, C next to G, A next to G, uh-oh, I made a mistake. I typed the wrong letter. I now have an A next to a G. Okay? DNA polymerase is going along 5' to 3'. All of a sudden, there's an A next to a G. Exonuclease raises its ugly head and goes backwards, chewing away nucleotides. Okay? That's what it's doing. It's destroying those nucleotides And in doing so, taking out that mispair that was there, the A paired to the G. So proofreading is removing that. Now, it doesn't destroy the whole strand. It just goes back for a few nucleotides. The exonuclease goes away, and the polymerase takes over and does its thing. That's pretty cool. We can describe that proofreading activity by its actual functional activity. So I'm going to remind you of it again. Here's the polymerase moving along, 5' to 3'. Oops, I put the wrong thing in. Exonuclease comes backwards. So if the polymerase is going 5 to 3, that must mean the exonuclease, which is what this is, the proofreading activity, the exonuclease is going 3' to 5'. You might wonder, why do we call it an exonuclease? An exonuclease describes an enzyme that starts at the end of a strand and moves backwards. Or moves inwards, we think of it that way, okay? There's not a backwards and forwards per se. In our case, backwards is counter to the replication, okay? All right, so now one of those activities makes sense. By having a proofreading activity, DNA polymerases improve their accuracy of copying a strand by about 1,000-fold. Proofreading gives about 1,000-fold improvement over an unproofread DNA polymerase. And yes, there are DNA polymerases that do not have proofreading. We see them associated commonly with certain viruses. HIV is one of them. The DNA polymerase that's in HIV, we'll talk about later, is a little unusual. One of its unusual things is it has no proofreading activity. Therefore, it makes a lot of mistakes when it's going along, which is bad if you're a cell, but it's very good if you're a virus that's trying to evolve away around drugs that are trying to stop it. More errors are good. You make a few strands that aren't viable, okay. Maybe you make 100 that now will survive the drug. So errors make good sense for a virus in particular. Did you have a question?
Exonuclease and DNA polymerase are the same enzyme?
Yes. So his question is exonuclease and DNA polymerase 1 the same enzyme? What I'm describing to you are 3 enzymatic activities in the same protein that we call DNA polymerase 1. So DNA polymerase 1 is the name of the protein. The 5' to 3' polymerase activity is one of the functions of that protein. The 3' to 5' exonuclease is another function of that protein, a catalytic function, obviously. They both are catalytic functions. And the third I'm getting ready to describe to you. But before I describe the third, any questions on what I've been telling you so far? Yes, Megan.
So does it happen simultaneously that the enzyme is adding nucleotides and it's checking to see if it did it right, a few nucleotides back?
Okay, so our question is the checking going on simultaneous with the addition? And the answer is sort of. Okay? So what can happen is that when you could imagine if you put an A across from a G, you got a little bit of a bulge. Okay? And that bulge may pose a barrier to going any further, or that bulge may not be realized until the enzyme itself gets a little bit further. Okay? So it's not a perfect scheme. So if the bulge is recognized, let's say in the first few nucleotides, then the polymerase has a chance to come back and chew it in. If it's not recognized and the polymerase just keeps going along its merry way, then, in fact, that mistake will be incorporated into the DNA, and there's other things that have to be done to fix it. Does that answer your question? Okay. Okay. Now, I said 3 activities. I've told you about 2. The third is found in DNA polymerase 1. It's not found in any other E. coli DNA polymerase. This one is also an exonuclease activity. And this one works in the 5' to 3' direction. And you say, oh, well, more proofreading, right? No, it's not. Okay? Because if you think about it, the polymerase doesn't really have connection with the 5' end once it's left to it. Once it's left it, right? It's connected to the 3' end because it's just made that 3' end, but the 5' end's way back over here. Right? DNA polymerase 1's 5' to 3' activity actually removes the RNA primer. Now, at first, it may not be apparent to you how it does that. I'm going to show you that in the figure in a little bit. But suffice it to say that a primer has a 5' end. And if the DNA polymerase 1 grabs a hold of that primer, its 5' to 3' activity can chew it out on the front side, while on the back side it's replacing it with more new DNA. And I'll show you how that occurs. Okay? So the 5' to 3' activity is essential for removing those RNA primers. If you take that function out of DNA polymerase 1 of E. coli dies. Okay? That E. coli will die. You might wonder why. Well, there's a very good reason why. The very good reason is that if we had a DNA that we left little pieces of RNA in, it would be stable, it would be a double helix. But when we came back and tried to replicate it, DNA polymerases, normal DNA polymerases, will not copy RNA. So now we've got all these little segments that would be left out. The cell will die. Now there are DNA polymerases that will, in fact, copy RNA. They're known as reverse transcriptases. Reverse transcriptases. HIV is a reverse transcriptase. That is, HIV has, or I shouldn't say is, it has a reverse transcriptase. It has an enzyme capable of copying RNA and making DNA from it using the base pairing rules. DNA polymerase cannot do that. Okay, so I'm going to say more about those as we get going along. All right, so now we start to get an idea about what's happening with enzymes. And we see that DNA polymerase 1 is a pretty important enzyme. DNA polymerase 1 uses metal ions to perform the catalysis. You've seen metal ions do a variety of things. We've seen activation of nucleophiles. And in fact, what's happening in this particular reaction is there's a nucleophile, that's the hydroxyl group, is being activated to attack the alpha phosphate of a triphosphate. So if this is, let's say, DATP, let's think about what DATP looks like. DATP has sugar on one side, it's got the base up here, and then it's got 3-phosphate. Phosphate, phosphate, phosphate. Everybody picture that? Sugar, base, phosphate, phosphate, phosphate. The phosphate, phosphate, phosphate are all linked on carbon number 5. The phosphates are joined to each other. If we think about those phosphates, the one that's immediately joined to carbon number 5 is called the alpha phosphate. The middle one, that is the one next to the alpha one, is called the beta phosphate. And the third one, not surprisingly, is called the gamma phosphate. When a phosphodiester bond is made, the alpha phosphate becomes linked to the hydroxyl on the 3' end. That means that the beta and gamma are kicked out, and they're kicked out as pyrophosphate. Anybody remember from last term why that's important? The what?
They get broken down for the reaction.
They do get broken down in a subsequent reaction, and that helps to pull the DNA polymerase reaction forwards. Okay? Very cool. All right. So that's what's happening during the catalysis, that is the making of the phosphodiester bond. Okay. Minor groove interaction. This is just showing you how some of the amino acids are reading those hydrogen bonds I talked about last time that are in the minor groove of the base pair between the DNAs that are between the strands that are there. And what else do I want to show you? I want to show you primase. So I described a situation before where if I put this strand out there with DNA polymerase and 4 DNTPs all by itself, nothing happens. It requires a primer. The primer in the case of a cell is an RNA primer. You see it's anti-parallel. 5' to 3' that way on the top strand. 5' to 3' this way on the bottom strand. New nucleotides will be added to the 3' end and will be extended in this direction, and we can see that happening right here with the blue. Okay. Now, it turns out cellular replication it always is this way, is more complicated than that. When DNA replication occurs, it's very easy to think, oh, well, that last picture, all I had to do was get on there, start at 5' end, move 3' end, I've got my DNA molecule. And if all I'm replicating is one strand, that's fine. But cells don't have to replicate one strand. They've got to replicate both strands. When they replicate both strands, they run into a logistical problem. And the logistical problem is this. Here's starting above this 5' to 3' that we were talking about before. As long as I just keep moving this guy along, it just keeps going. Right to left, no problem. How do I replicate this guy? It turns out cells replicate this strand almost simultaneously with this strand. Almost simultaneously. Well, why is that a problem? Well, this strand has got to be replicated also in the 5' to 3' direction because that's the only direction in which DNA polymerases work. It's 5' to 3'. So, well, I've gone a certain distance, and remember that DNA polymerase can only work on a single strand. If this guy is pointed 5' to 3', that means this guy's got to go this way. So as this is going this way, that has to be going that way. That means that these two strands are replicated in fundamentally different ways. The leading strand, which is the top one, is synthesized in a single piece. It starts and it just keeps going and 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 going, nothing to stop it. Remember we've got, and I'll describe in a bit, we have to unwind the strands in order for replication to occur. This strand can only start replicating as a new section of it gets opened up. A section gets opened up. Oh, I can put a primer on here and do it. Then I wait for the next section. And then it gets opened up and I put a new primer on and I do it. The bottom strand is called the lagging strand because it has to wait for that unwinding to open up a new section for it to replicate. That means that the bottom strand, the lagging strand, is made in pieces. Little tiny pieces. The pieces have a name for the scientists who discovered them. They're called Okazaki fragments. And virtually every cell on the face of the earth uses this mechanism. Leading strand in one piece, lagging strand in many pieces. Now, what do you suppose is on the end of each one of these pieces? A primer. Okay? Let's think about DNA polymerase 1. DNA polymerase 1 finds a primer, it gets replicating, it goes along, and all of a sudden it hits RNA. The 5' to 3' polymerase goes, oh, no problem, I chew it out of the way. And the DNA polymerase continues doing what it's doing behind it. So that the RNA primer is removed by the DNA polymerase 1. Now, the DNA polymerase does not have the ability to join the end of this one with the end of this one. There's a little tiny gap between the two that's left. The DNA polymerase, once it's realized it's removed the RNA, the DNA polymerase goes flying away. And now we're left with a little tiny gap. That little tiny gap has to be joined together. The pieces have to be joined together, and they're joined together by an enzyme known as DNA ligase. So now we see another enzyme that has to be involved in replicating DNA. We've seen primase. We've seen DNA polymerase. We've seen ligase. There's more. Oh boy. The proteins are really cool. Yes, Carl.
So the gap between the two, is there missing nucleotides, or are you just saying it's the two right next to each other?
Okay, good question. So the gap is not missing nucleotides. What it is the 3' hydroxyl is sitting right next to the 5' phosphate, but they're not joined. Okay? That's what it is. But there's no missing nucleotides. It's just those two things haven't been covalently linked together. Okay? Good question. About how long is each Okazaki fragment? It will vary a bit, but a few hundred nucleotides. Now, okay, so that's a schematic about what happens here. Let's think about some more things that have to be present. I told you the other day that we have to untangle these strands, and up ahead, if we don't take care of that untangling, we're going to create a knot. So you know that somewhere up here we've got DNA gyrase in E. coli, which is a topo 2 isomerase. So we have to have that. That's another piece of the puzzle. What's taking the strands apart? DNA polymerase can't do it. DNA polymerase will not take apart the strands. So there's another enzyme that has two different names that does it. One, the most common name is Helicase, Helicase. Helicase is also called DNA B. That was how it was originally, its original name when they isolated it. It's now more commonly called DNA helicase. Okay? DNA helicase's job is simple. Pull the strands apart. Well, pulling strands apart takes energy. So DNA helicase requires ATP to do that. And what DNA helicase does is nothing short of remarkable. I want to run some numbers by you. Okay? You don't need to know these numbers. The E. coli genome is about 6 million base pairs in length. That entire 6 million base pairs can be replicated in about 20 to 25 minutes by two enzymes, or two replication forks, starting here and going all the way around and doing that replication in about 20 to 25 minutes. If you do the math, you discover that that replication fork, which we see right here, that replication fork has to be moving at 1,000 nucleotides a second. 1,000 nucleotides a second. That's pretty remarkable, especially when you consider that this replication machinery makes an error about once every million or 10 million bases. 1,000, imagine typing 1,000 characters a second. Okay, pretty cool, right? In order to have this replicate 1,000 nucleotides a second, DNA helicase has to be pulling apart the strands at the rate of 1,000 nucleotides a second. That is, if you think about 10.5 base pairs per turn, that means essentially about 100 turns per second. Well, if you look at your car and your engine is turning at 100 times a second, you're doing 6,000 RPM. DNA helicase is operating, I'm not exaggerating, at 6,000 RPM. That's pretty mind-boggling. No wonder it needs ATP. 6,000 RPM. If it can't keep up that pace of pulling the strands apart at that rate, DNA replication will slow and the cell will be in trouble. So, DNA helicase is another important piece of this replication puzzle. There's others. Another one is the fact that, well, DNA helicase can pull these strands apart, but there's usually a little bit of a gap between the place where it pulls the strands apart and the place where the DNA replication is going on. At this point, the DNA molecule is in a very tenuous state. It's susceptible to damage. If I nick one of the strands by a chemical process, I've just broken the DNA molecule and it can't be fixed. So cells protect those single strands using a protein called SSB, single strand binding protein. So single strand binding protein comes along when it finds a single strand of DNA and it covers itself with it, protecting it from damage. Okay, now we've seen essentially all the players except for one. And I've left it out on purpose to tell you about it. When Arthur Kornberg isolated DNA polymerase I, he thought he had the answer to the DNA replication puzzle. It replicates DNA. It's got the activity that we need. It even has proofreading activity. This is all very cool. And then they discovered that DNA replication occurred by the way I described to you. Two replication forks. Not 100, not 200, but two. They start at a specific place, and they go outwards, and then meet elsewhere on the circular bacterial chromosome. Remember, bacteria have circular chromosomes. Okay? Well, this was a problem. Because DNA polymerase I, when you put it onto a double-stranded DNA, would go on for about 100 or a couple hundred nucleotide base pairs, and it would fall off. And they knew it was too slow. If it kept falling off and its rate of replication, it couldn't possibly replicate that DNA in the time frame they knew it was being replicated. Something's wrong. And there was something wrong. They had the wrong DNA polymerase. Now, DNA polymerase 1 does have a role. The role is dealing with the Okazaki fragments. But it's not the main polymerase that replicates the bacterial chromosome. The main one, the reason they didn't discover it at first was there's only about 5 or 6 copies of it in a bacterial cell. Well, there are thousands of DNA polymerase 1s. That seems odd, but not really. Think how many Okazaki fragments there are. The more DNA polymerase 1s you have, the more likely you are to find the Okazaki fragments and fix them. And you only need 2 the other DNA polymerase to start and then go outwards like this. So 5 or 6 made a lot of sense. But they had to grow up a lot of protein to find that other DNA polymerase. The DNA polymerase that they found that has that replicates most of the E. coli chromosome is called DNA polymerase III. It has the same basic properties as DNA polymerase 1, except it does not remove primers. So it has a polymerase activity that goes 5' to 3'. It has an exonuclease activity that serves as a proofreader that goes 3' to 5', but it does not have anything to remove primers. Now, DNA polymerase III turns out to be a much more complicated enzyme. It has multiple subunits, and the multiple subunits have individual functions. I'm only going to talk about a couple of them. Okay? So if you'll bear with me, I'm going to talk about DNA polymerase III, and then we'll have a song. How's that? Okay. This is what DNA polymerase III schematically looks like. Okay? It turns out that DNA polymerase III is a dimer, meaning it has 2 duplicate sets of replication and exonuclease functions. This whole thing can replicate. This epsilon right here is the proofreading part. This holds everything together. Okay? And there's DNA helicase ahead of everything, unwinding the strands. This guy right here turns out to be the key to why DNA polymerase III is fundamentally different than DNA polymerase I. So far, the main difference I've told you has to do with the not being able to remove the primers. That's not the key difference. The key difference is that DNA polymerase III, when you put it onto a DNA, it goes on and it stays on. And it replicates at 1,000 nucleotides a second, a pretty critical thing. DNA polymerase I doesn't work that fast. So why is DNA polymerase III fundamentally different? Well, let's imagine. Here I have a double strand. Okay, I'm moving along. I'm peeling it apart. Here's my DNA polymerase I. It goes along for a little ways, and it falls off, as I've described. It'll go for a couple hundred nucleotides, and it falls off. Falling off is good, because we don't want it chewing up with its exonuclease activities a lot of other stuff that's there. We have it fall off. That's good. DNA polymerase III, we want it to get on and stay on. Well, how do we keep it on? It turns out we have a little ring. This little ring goes around the strands being replicated. It holds the DNA polymerase onto the DNA. This ring is called a beta clamp. Beta clamp. And what it does is it anchors the polymerase activity right onto the strand being replicated, and as a consequence, the polymerase does not fall off. DNA polymerase I does not have a clamp. It goes on, it falls off. When we look at human DNA polymerases, they also have a lot of complexity, but the common feature that they have is they also have a clamp. In human beings, it's called pCNA. pCNA. But it's equivalent to the beta clamp, and it has almost identical structure. That is, it's a ring that holds onto the DNA. Okay. Now, So, schematically, here's a core that holds everything together. Here's the very front of the enzyme moving forwards, where the DNA polymerase is peeling apart the strands. I'm sorry, DNA helicase is peeling apart the strands. Here's the polymerase section with the proofreading, which is the epsilon subunit. And here's the other one. Well, why do we need two? Remember that both strands are being replicated simultaneously. One is doing the leading strand, as the other one is doing the lagging strand. Yeah, it's very hard to get your head around. And I'll tell you it's so hard to get your head around. I'm going to show you a figure or two, or actually an animation, that the animation doesn't even do it very well. Okay? It's very hard to get your head around. But before we go into that, let's do a song. What do you say? That'd be a good break? Okay. Well, the song I have for you is one I wrote for this class last year, and it's about the structure of DNA, and it's to an old Simon and Garfunkel tune. If I can find it. There it is. And I hope you know it. It's an old song called Feelin' Groovy. And it's called Major Groovy. Okay. The DNA forms A and B. Please join. Have bases complementary. Despite the similarities, they differ in their major groovies. Na 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 na. Major groovies. Transcription factors with their binding cause DNA to start unwinding, holding it aggressively by forming bonds in major groovies. Na 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 na. Major groovies. For proteins, the key to sequence ID is hydrogen bonding, each base pair unique. Purine, pyrimidine, patterns discrete, and DNA's most major groovy. Na 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 na. That's it. Major groovy. Okay, it's a short one. Okay, now I've thrown a lot of different mechanisms and so forth at you. So what I want to do is try to show you some videos that try to capture the complexity of this, and I will tell you they don't do a real good job. But in some cases, I think they are helpful. So here's one. These are slow. All right. So here's the simple replication fork that we saw before. We know it goes 5' to 3'. Here's the leading strand being replicated. The DNA polymerase is sitting here. And remember, we've got the other side. Here's the RNA primer. And this other side has got to now look ahead of the primer because it's got to go this way as the other part of it is sitting over here. That's a logistical problem. It turns out that this piece actually will loop out. It's not shown in this figure in order to accommodate what's necessary there. So now we see, okay, what's happening here. Once the strand is made, it starts rewinding. So we're seeing double helix forming. We're seeing this leading strand being made. Okay? Here is what's going on with the proteins. And there we go. Here's your helicase. Now, this figure is actually wrong. Okay? It shows DNA polymerase as one enzyme over here. This is polymerase 3. And it shows another one coming in from elsewhere and going this way. And that's not what happens. Okay? You'll see it in a second. Come on. Well, maybe we won't. Okay, so here comes our primer. Now watch, you'll see another enzyme come in, which is not right. Come on, 1,000 nucleotides a second. Okay, so that is an incorrect depiction. Oh, sorry, didn't mean to do that. All right, now let's look at what actually happens. At least they attempt to show you what happens in 3 dimensions. Now, remember I said that the polymerase has got to adjust for the fact that it can't just stretch way ahead and get that primer. Instead, the DNA strand bends out and forms what's called a trombone. Now, let's try to see what's happening here. In fact, they're trying now to show you, here's one side of the DNA polymerase 3, here's the other side of the DNA polymerase 3, leading strand going this way, lagging strand going this way. Okay? Here's our primer. Okay? And now look. Now we can replicate. It will. There we go. Okay, so we see replication now going on from the primer. Now, this strand is a lot more complicated. We see several things that have to happen. Leading strand replication occurs faster than lagging strand does. So what that means is this thing grows, and then it shrinks, and it grows, and it shrinks, just like a trombone. And this guy, the leading strand, has to occasionally wait for this guy to catch up. Kind of cool. Okay, that's the general idea of what we're talking about during E. coli replication, and very similar things happen in our replication system as well. Okay, this has some audio, I believe.
When DNA replicates, its strands are separated by the enzyme Helicase. Single-stranded DNA binding proteins keep the strands from reannealing. One DNA strand encodes the leading strand, which forms from its 5' to its 3' end, using Dna Polymerase 3. No problem here, but the lagging strand presents problems. It has to form from 5' to 3' too. It forms in pieces called Okazaki fragments. First, an RNA primase lays down an RNA primer. Then, Dna Polymerase 3 lays down new DNA. The process repeats again and again. Dna Polymerase 1 replaces the RNA primers with DNA. Finally, DNA ligase links the Okazaki fragment.
All right, so you get the idea here. Now, I've linked several different videos there. The ones I've just shown you here are the best ones, with one exception. And the one exception is the inner life of the cell. It has really very little to do with DNA replication. But if you've never seen this video produced at Harvard University, you should take a look at it. It's absolutely beautiful in terms of what it does. I hear everybody rustling. It's Friday. Let's call it a week. And I'll see you guys on Monday.